Okay, so we are going to talk about a process called transcription. So we said that the central dogma of molecular biology says that we have double-stranded DNA, and this DNA is the genetic material. Sometimes it's called the genetic code. DNA that codes for a protein is called a gene. So this gets a little more complicated, but for right now, we're going to say all of this is the DNA. So the next thing that happens is this DNA, this double-stranded DNA, a copy of it is made into single-stranded messenger RNA. This process from double-stranded DNA to single-stranded messenger RNA is called transcription. Sometimes you'll hear this messenger RNA called a transcript. Ultimately, this transcript is then going to become translated into protein. The point of all of this is that our DNA codes for proteins, and we have to go through this intermediate step of the mRNA transcript. It's like if you had a letter written in Spanish and you had no way to translate this letter from Spanish to English, so you had to translate it from Spanish to French first and then from French to English, for example. There's no direct way to go from DNA to a protein. You have to go through this intermediate step of transcription where we're taking double-stranded DNA, converting it into single-stranded messenger RNA. So remember all the differences between DNA and RNA. We'll talk about the different types of RNA here in a little bit, but remember, DNA is double-stranded. Messenger RNA is single-stranded. DNA, the D stands for deoxyribose, meaning that there's just a hydrogen on that 2' carbon on the ribose. RNA is ribonucleic acid. Ribose has a hydroxyl group on the 2' carbon. So I would expect you to be able to differentiate between mRNA and DNA because RNA has a different sugar than DNA. RNA is single-stranded. DNA is double-stranded. And they use different nucleotides. DNA uses the nucleotide thymine. mRNA uses uracil. Okay, so we are going from DNA to RNA. First, let's just say what the types of RNA are. There is mRNA. M stands for messenger. It's the message. It's this transcript. There's rRNA. R stands for ribosomal. Ribosomes are actually made up of rRNA, and we're going to talk about this next because the ribosome is the cellular location where translation happens, the next step. And then the last type of RNA is tRNA, and T stands for transfer. And we're going to see where the tRNA is involved also in the next process that we'll talk about, translation. So messenger RNA, M, and you'll see that these letters in front of the RNA, the R, N, and A are capitalized, and the letters in front are lowercase. So these are different types of RNA. Again, just a little reminder, I know people are coming to this class with lots of different levels of biology. If this is all brand new to you, please watch the videos that I've posted that are kind of a crash course in replication, transcription, and translation. A lot of this is leaving out a lot of the more elementary reactions that are happening. Okay, so transcription. Let's talk about where it happens. So remember, we're going from DNA to RNA. So the DNA is only ever in the nucleus. So let's draw a cell here. Let's say, I'm going to draw it over here. Here's a cell. Here is the nucleus. And then I'm going to draw this. On the outside of the nucleus, kind of continuous with the nucleus, you'll see all of these when you look at pictures, this structure that kind of has these back and forths. This is called endoplasmic reticulum. Most of the time it's just called ER. There's two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum is rough because it's studded with ribosomes. So I'm going to draw these little dots all over the endoplasmic reticulum. And what these little dots are the ribosomes. So if there's a ribosome on the endoplasmic reticulum, it makes it rough endoplasmic reticulum. Smooth endoplasmic reticulum has a different job in processing and does not have the ribosomes on it. Okay, so this is important. So that we have the nucleus. The nucleus is where the double-stranded DNA lives. So I'm going to draw here some DNA. In my nucleus. What happens is the enzymes that undergo transcription are in the nucleus because the DNA never leaves the nucleus. So this process of transcription, the physical process, is inside the nucleus. But it's not done there. The transcript then leaves the nucleus. So I'm going to write DNA transcribed to messenger RNA inside the nucleus. The next thing that's going to happen is the messenger RNA is going to leave the nucleus through a nuclear pore. So there will be these little pores all through the nucleus. Messenger RNA is allowed to leave the nucleus through a nuclear pore. DNA is not. DNA never leaves the nucleus. This is why we need this copy of messenger RNA to get it out of the nucleus. The nucleus is the safest place, and we want to keep the genetic material for this cell as safe as possible, so it never leaves the nucleus. Once the messenger RNA leaves the nucleus through a nuclear pore, it finds a ribosome. And on the ribosome, this is where translation happens. And this is the next step. So transcription physically happens in the nucleus. The next step that we'll talk about later, translation, happens on the ribosome, which is right outside the nucleus. So the transcript, the messenger RNA, is allowed to leave the nucleus where the DNA is not. So that's the first thing that we need to talk about. So let's talk about the stages of transcription. So how do we know? So another thing that's important to point out is that when we undergo DNA replication, we are replicating the entire genome. We're taking all of the chromosomes and we are replicating from beginning to end. We're doubling all of the genetic information. In transcription, we only want to copy a segment. Let's say here's the gene from here to here. So we only want to make a transcript of this part because this is what's coding for my transcript, which is eventually going to become my protein. So transcription is starting at a specific point and it's stopping at a specific point. It's starting where the protein starts and it's going to end where the protein ends. So the first thing that needs to happen in transcription is we need binding of the RNA polymerase at the promoter site. The promoter is a site on the DNA that's going to promote transcription. This is where the RNA polymerase is going to bind. RNA polymerase is actually it's called a holoenzyme, means it's made up of a lot of different enzymes. This RNA polymerase is recruited to the promoter by something called transcription factors. Transcription factors recruit the RNA polymerase to the promoter. Now, something that's important to remember here, these transcription factors, most of them will have a zinc finger motif, meaning the shape of the transcription factor is shown. It has a zinc finger. The zinc is a positively charged metal ion. What's important is this zinc finger, it binds to the DNA. It doesn't really bind, it's attracted to the DNA because remember the DNA has that sugar phosphate backbone. And remember what phosphate is. Phosphate is very negative. So this is an example of a non-covalent interaction that is important in these processes of DNA transcription. The transcription factors being recruited to tell the polymerase where to start transcribing this gene are recruited by transcription factors. Transcription factors stay on the DNA because they are positively charged. This is a non-covalent interaction with the DNA so that these zinc fingers can stay on the DNA. So always be looking for examples of covalent and non-covalent interactions. So covalent interactions for DNA is the phosphodiester bond is covalent. Non-covalent would be the hydrogen bonds between the bases, the complementary base pairs, those are hydrogen bonds, and this would be another example of a non-covalent interaction. So once the transcription factors are recruited to the promoter, the RNA polymerase comes to the promoter site and will start transcribing. So this is the same as DNA polymerase. RNA polymerase can only add new nucleotides to the 3' end of the growing transcript. So remember the directionality of the strands 5' to 3'. RNA polymerase, like DNA polymerase, will only add a new nucleotide at the free 3' hydroxyl. So there's different sites in this, what is the gene, what is the DNA strand, and what is getting copied is called the template strand. So the place that is being copied, the DNA that's being copied, is called the template. And that's the strand that's being copied. There are also a cluster of genes often. So genes for certain pathways are usually coded next to each other on a strand of DNA. So for example, if you are undergoing glycolysis, the 10 enzymes that undergo glycolysis are all going to be coded for in the same area of DNA. So then when transcription starts, it can just continue to transcribe however many proteins or enzymes it needs. So what happens, similar to the replication fork, in replication is there's a transcription bubble where these hydrogen bonds between these complementary base pairs are going to break, and the RNA polymerase will come in and it will use the template strand to start making the messenger RNA transcript. Again, there's lots of good pictures in the lecture and also a good video that I've posted on transcription. A lot of these examples are this whole process here of the RNA polymerase binding, this is called initiation. The next step would be elongation, which is where the DNA is transcribed until it reaches the sequence that tells us when we are going to stop, which is called a termination sequence. And then the last step is termination. So initiation, elongation, and termination. So this is a much more complicated process in a eukaryotic cell versus a prokaryotic cell, just like all processes. In a eukaryotic cell, there are ends that are put on to this messenger RNA to kind of keep this transcript safe. So remember, this transcript, once it's made, is it's going to leave this super safe environment of the nucleus and go out to the ribosome. So sometimes after transcription, in prokaryotic cells, these primary transcripts also can be called pre-messenger RNA. The primary transcripts, so I'm going to write pre-mRNA or primary transcripts, are capped and methylated. And this has to do with protecting this transcript once it leaves the safe environment of the nucleus. So it's capped by an enzyme called guanylyl transferase at the 5' end. So it caps the guanines at the 5' end. So the 5' end is capped with a guanine. And the 3' end gets what's called a poly-A tail, where it essentially just adds a long string, sometimes up to 100 to 200 adenine residues at the end. This is called polyadenylation. We don't exactly understand why this happens. It is thought to provide stability to this transcript, so when it leaves the nucleus, if anything was to happen to these transcripts, it would be at either of these ends, and it would interfere with the capping or the poly-A tail, but not necessarily damage the transcript. And then the transcript is going to go ahead and be translated. Okay, so we have started with double-stranded DNA in the nucleus. Initiation factors, transcription factors are going to bind to the promoter. It binds with the positive charged zinc fingers in the transcription factors to the negatively charged sugar phosphate backbone of the DNA. That tells the promoter where to start transcribing. It transcribes till it reaches a termination sequence. Then this primary transcript is capped and polyadenylated to protect it, because the next step that's going to happen is this transcript will now leave the nucleus to become translated into protein on a ribosome.